Well, thank you so much. For starters, I should say that I'm not a neuroscientist or even close to it. I did marry one, but yes. Well, and my wife tells me that too. I'll mention her later. I'm actually a chemical engineer. But when Bank asked me to speak at this, these are just fantastic symposium, and I was just honored to do it. And I'm going to try to explain how chemical engineering in a couple ways could relate to neuroscience as I go through the talk. But I thought what I'd try to do is I thought about preparing this talk, since there are many high school students, I was trying to put myself back in your place and think about what might be useful to go over. And I thought I'd actually start out and tell you just a little bit about my own career and how I got to be here today, in a sense. So I'll just start when I was a few years older. So when I went to college, I majored in chemical engineering. And when I got done, there was this big oil shortage or gas shortage in the United States. And what happened is the prices of gas at all these gas stations kept going up and up and up. But the thing is if you were a chemical engineer, what that meant is you got a lot of job offers. And I remember myself, pretty much every one of my classmates, that's what they did, is they went into the oil industry. And so I thought I should do the same. I applied to all these oil companies, and I actually got 20 job offers. Four actually from Exxon alone. And it wasn't like I was that good or anything, but it's just that they had a lot of job offers. So, when I went to these interviews, I wasn't very excited about it. What they were talking about was increasing the yield of oil or some of these chemicals by about 0.1%. And I really wanted to, I felt, make more of an impact than that. I wanted to do something that I felt would really help people. And one of the things I did when I was in high school is when I was in college, one of the things I did is I helped start a school for poor children in Cambridge, which is where MIT is. And one of the main things I did is develop new kinds of chemistry curricula to try to make chemistry more interesting. And I love that. And one day I saw an ad, actually, to be assistant professor at an American college to do just that. And I wrote them a letter applying for the job. But they didn't write me back. But I really liked the idea, so I found all the ads I could where you could do something like that. I wrote to about 40 colleges to apply for being assistant professor to do that. And actually, none of them wrote me back. I guess even if you went to MIT and you weren't in I wasn't in like the chemistry education box, so I guess you don't get a letter back. So anyhow, I started to think, well, what other ways could I use my education to help people? And so I thought about medicine. So I wrote to a lot of hospitals and medical schools, and they didn't write me back either. But then one day, one of the people in the lab where I was said to me, Bob, he said, there's a surgeon in Boston named Judah Folkman. And he said, sometimes he hires unusual people. He thought very, very highly of Dr. Folkman. I won't say what he thought about me. So I'm going to start and actually tell you I'm going to give you a few examples of some of the things we've did. But this is actually a picture of Dr. Folkman's idea. It's actually from the New York Times many years ago about how his vision of how tumors grow. And what he said is a tumor cell becomes abnormal. It grows to about maybe a millimeter cube. But then it runs into what we call a nutrition problem. Cells in the center can't get nutrients or get rid of waste. So they die. So it couldn't get larger than that, except for the fact, what he said, is that somehow the tumor secretes a signal. He called it TAF, tumor angiogenesis factor. And see, normally you have blood vessels and they just hang around and don't do much. But what he says is in the presence of the tumor, what happens is all of a sudden these blood vessels, they start to multiply, they grow to the tumor. What it is, there's a particular cell type called the endothelial cell, and they start multiplying and grow right to the tumor. And that solves the tumor's nutrition problem because it gets the blood supply. And that causes a second phase of growth. And see, when this happens, the tumor gets bigger and bigger and ultimately can spread through these blood vessels. That's a process called metastases, and eventually it can kill. By the way, brain tumors are amongst the most vascularized tumors in the body. At any rate, one of the things he said was, if you could stop angiogenesis, maybe that would be a whole new way of stopping cancer. So when I came to his lab, actually, what he asked me to do, this was a theory, so what he asked me to do is, could I actually isolate for the first time a substance that could stop blood vessels from growing? And so how do you think about a problem like that? How do you think about finding a new factor? So we kind of broke it up into two parts. The first part is, where could you ever find something that might stop blood vessels from growing. And this is just a proof of principle at this point. So one of the things we thought about is cartilage. Cartilage is in your nose or your knee, and it doesn't have blood vessels. So I started to get, from the little rabbits we had in the lab, I got some of their cartilage, but I couldn't get very much. So, one of the things you learn in chemical engineering is scale up. So I went to cows. And I got some cow bones at a slaughterhouse, but it's like 1 or 2 bones. It wasn't that good. So I found out, where do all the bones go? And I found out they went to all these meat packing places. And I made arrangements with them to get all their bones. And I'd bring them to the lab, 3 or 4 times a week, and I'd scrape the meat off. Here's a picture of one of them. I'm actually quite proud of this. Normally there's a good bit of meat on, but I'd scrape it off, and then and I do this with thousands of bones. I mean, I spent 40, 50 hours a week doing this beyond all the other things I'll tell you about. But anyhow, but I got pretty good at it. But at any rate, you'd slice this off, and then I'd work out ways to extract it with different solvents like guanidinium hydrochloride, and then I'd purify the different fractions. So at the end of a couple of years, I'd have hundreds of different things I wanted to test. That brings up the second problem. How do you test for a substance that could stop blood vessels from growing? And if you look back at the history of medicine and biology, one of the things you see when people try to come up with different factors is really what's often rate limiting is the bioassay. And there was none. There was no bioassay for studying angiogenesis. And it's not easy to do because there's always background blood vessels wherever you could think about in the body or any organism. So we were trying to think, how could we create a bioassay? And one of the things we thought about was actually the eye of a rabbit. We picked it for a couple reasons. First, it's actually fairly easy to see, and rabbits actually have big eyes. You can look at them through an ophthalmic microscope. But also, what we found is that tumors, certain types of tumors, will cause blood vessels to grow from the edge of the cornea, and I'll show you this later, over about a 2 to 3 month period. And in that time, you could actually study, it's almost like a graph. There's no background blood vessels in the cornea, and you could have this graph, and you could follow how fast the blood vessels grow in the presence of different substances. But see, what we'd need is all the molecules in cartilage were fairly large molecules, and like proteins, proteoglycans. So what we'd need is a little polymer that we could put right next to the tumor that would slowly emit any of the factors that I'd isolated. So that was actually a key problem. Could we come up with a polymer that would not cause harm to the eye and that could release molecules of any size? And that was very challenging. That had never been done. And actually, Dr. Folkman, this was 1974, he was on the advisory board of the one company in the world working on materials that could release drugs, Alza. They had only, at that point, you could only release a couple molecules. But what they said was that you can't do it. In fact, Paul Flory was a Nobel Prize winner on their board with Dr. Folkman. They all said it's impossible for large molecules to get through the polymers. They said it's kind of like any of us walking through a wall. I mean, you could make it very porous, right? But then you're through the wall instantaneously. So the challenge is, could we come up with a way to deliver molecules of any size over periods of time? In fact, the literature said pretty much the same thing. Here's Chemical Engineering News in 1977 saying the use of polymer matrices for slow-release systems has been virtually restricted to small molecules. Really, the only thing I had going for me is I hadn't read any of this. So I spent 2 or 3 years in the laboratory experimenting almost Edisonian-like with different techniques. And I found actually over 200 different ways to get this to not work. But eventually, I was able to make little microspheres. Here's a picture of one. Here's one cut in half. In fact, now we also make nanospheres and all kinds of things. And what we showed is that we published a paper in Nature in 1976 showing for the first time you could release molecules of virtually any size for over 100 days and get bioactivity. Later on, we worked out ways to even get constant release. So one of the things, that you have to do if you're in academics, you end up giving presentations like this. So in 1976, I got asked for the first time in my life to give a presentation at a scientific meeting. And it was, and it was to a group of much older, that was many, many years ago. So I was, a lot younger, had a lot more hair and stuff. And so this was to a group of very distinguished chemists and chemical engineers. So the only talk I'd ever given before that was in 8th grade. And that talk didn't go real well. I remember in 8th grade what happened is I had to give this minute-and-a-half speech, so what I did is I wrote it out, and the night before, I stood in front of my parents' mirror for 3 hours just reciting it over and over again. And I did okay, in front of my parents' mirror. But the next day, I got up in front of my 8th grade class, and I started to recite the speech. I should have brought the sheet of paper up, see, I'm doing this today. But I didn't, and so, but still, for the first minute and 2 seconds, I did okay. I kind of just recited it. And then I could not remember the next word. And I stood up in front of my 8th grade class for another minute, just absolutely frozen, and saying nothing until my 8th grade teacher finally told me to sit down and gave me a not particularly good grade. I think it was an F. So ever after that, I practiced for, really long times and so forth. So now, many years later, I had to give this talk, in 1976 to a very distinguished group, like I say, of polymer chemists and engineers. So I practiced it for a couple of weeks. And when I got done with this 20-minute talk, I thought I did much better. I didn't forget too much what I was going to say. I didn't stammer too much. So I thought when I was done with this talk that these older chemists and engineers, being nice people, would want to encourage me, this young guy. And when I got done, I stepped off the podium, and a whole bunch of them came up to me, and they said, "We don't believe anything you just said." They said, just like the ALSA people said, that the large molecules can't diffuse through solid polymers. We were also using organic solvents, and they said that's going to destroy lots of the substances that you put in. And actually, things got worse after that. I remember my first 9 grants that I wrote to the government to try to fund this work got turned down. And even at MIT, when I became a professor there, after the first year or two, the department head who hired me, Nevin Scrimshaw, he left. And so then they had different associate department heads, and they both decided to sit down with me at different times and give me advice. And basically, their advice was to look for another job. But the key thing in science, as you know, is people ultimately repeating what you do and so forth. And what would happen is, starting by 1979, different research groups started using what we did, and then the question shifted. And I'll get into some of that, but to how could this work? And to understand it, what we did, is we cut thin sections through these polymers with a cryomicrotome. And see, here's a 5-micron thin section, and it turns out, see, a molecule even 200 molecular weight or greater can't go from one side to the other. But if you put a drug in it, we'll put a red one in it just so you can visualize it, then you see what we call a phase separation. So you see the polymer here, drug here. But if you release this, say, for a year, and then you cut a thin section, what you see is, in sharp contrast to what you saw before, are these pores. These pores didn't exist 2 slides ago, so they're made by the way we develop these. And these pores are large enough so that molecules, even millions of molecular weight, can get in. But what happens if you do serial sectioning or various image analysis, which we did, you see that there are tight constrictions between the pores, and the pores are incredibly winding and tortuous. So it takes a really long time for the molecules to wind their way through. One way I explain this to people living in Massachusetts, I don't know if people have ever been there, but if you've ever driven a car through Boston, it's very similar to this. It's actually, there's a term we have in chemical engineering called tortuosity, and this in Boston have very high tortuosity. And over the years, our students have worked out ways to regulate that tortuosity by various means, and I'll just sort of highlight some of them, like you can change the particle size of the drugs, you can change the amount of the drug, because that changes the interconnections, you can coat it, the system, you can change the molecular weight of the polymers. In fact, one of the things, so what we've learned over the years is we can actually make these systems very slowly release, very quickly release, orders and orders of magnitude. And our students, I won't do the math today, but we've even created mathematical models whereby you can predict all these things. So having developed this foundation, now we were in a position to address different things. So I'll just give you 3 different things that we tried to do. First, of course, is why we started this, angiogenesis. This was the bioassay we wanted to develop, and now we did have these polymers. So what we did is, now I had all these different fractions that I wanted to test, so we actually did 2,000 rabbit eyes. 1,000 rabbits. And I should say almost every fraction we tested did not work. I kind of think of those as the controls. There actually are really controls if you think about it, because they're purified, the same source, it's from the same system, just purified slightly different. But one of them did work, and I apologize if people don't like the sight of blood, but it's the only way to show this. So I'm going to show you rabbit eyes, and this is about 8 weeks after the start of an experiment. And this is from a paper we wrote in Science in 1976, which is the isolation of the first angiogenesis inhibitor. So this is a tumor. It's a little bit cloudy. This is the blood vessels. Normally there were none, but you see the sheet of blood vessels growing over the polymer, that's where it was, on the way to the tumor. If you looked at this eye just 2 or 3 weeks later, it would be three-dimensional, out of the orbit of the eye. We sacrificed the animals before that. But when you put the inhibitor in, the vessels are sparser, they avoid the polymer, and about 60% of the time these tumors never grew. So we published this in 1976. It took another 28 years from this paper before the first angiogenesis inhibitor was approved by FDA. But starting, and in the end it was because of companies like Genentech and others, but basically they would use the different technologies we developed, but it just took, unfortunately, an enormous amount of time and effort. But starting in 2004, what we see was very exciting. You see one after the next angiogenesis inhibitor being approved by FDA. Avastin, which is approved for glioblastoma and others, is probably the #1 or 2 largest selling biotech drug in history. And not only are they used for all different kinds of cancer, you know, they keep getting approved, but for different eye diseases. Like before this, there was no drugs to treat what's called macular degeneration, which is hemorrhage, which you get abnormal vascularization in the back of the eye. So these were the first drugs that could start to treat that, drugs like Lucentis and Eylea, and basically it's even reversed macular degeneration in a number of cases. The second thing that happened, and I didn't even realize this myself, but you know, we published these papers in Nature and other places, and it turned out that actually a lot of developmental biologists and neuroscientists started using them to deliver different factors to study things in the brain. Because, see, normally if they just tried to give them in the body, they'd be destroyed right away. You couldn't study them. So now they put them in these little polymers that a lot of times people would call them LVACs or other things. That's like after the trade name. And actually what I found when I did some literature reviews is there's literally hundreds of labs, you know, and I was always surprised. I have these different neuroscientists coming to me and saying, could I get some of this LVAC stuff from you? But basically, I thought I'd just give you just a few quick examples of what people did. These are just different papers. Here's one group looking at studying, releasing a molecule to study eye-specific segregation. Here's another where they're releasing another substance to study a mammalian neural map. Here's another one where they're looking at growth in the rat visual cortex. This next one's actually particularly important to me. Here, this was actually a study out of Yale, and where they're studying things to look at developing spinal cord. The reason that ended up being important, this is a woman named Susan Hockfield, and she later became president of MIT. So I was very glad that she used some of the things that we did. She was actually a great president. And there's more controlled release of growth factors, toxins, and so forth. So all these things ended up, not with my intent, but people using them in neuroscience. The third thing that I want to do, again, being an engineer, you know, like I said to you in the beginning, I wanted to do stuff that I thought would ultimately help people. And the way I started to think about that is, could we ever use some of our discoveries to make products, not just angiogenesis inhibitors, but, you know, so many molecules that people take, either they don't take them or you try to inject them and they're destroyed in the body. So we wanted to do a proof of principle showing you could release molecules for long times. And here we took insulin and released it in a diabetic rat for over 100 days. So, you know, we did all this work. And one of the things that Dr. Folkman once said to me, he said, Bob, you know, we should file a patent. And in the history of Boston Children's Hospital, which is where I work, they actually never had a patent. It's a very good hospital, but they never had a patent. So we filed a patent. And 6 years in a row, the patent examiner turned it down. He wasn't that bright, but the thing is that it didn't really matter because he was calling the shots, not me. And finally, our lawyer said to me that, Bob, you should just give up. You're wasting a hospital a lot of money and so forth. But I don't like to give up. And I kept thinking, you know, how could I convince, you know, science wasn't working. And I started to think, how could I convince this guy, you know, legally, of course, that he should allow the patent. And as I mentioned to you in the beginning when we first did this work, as I said, all these people told us, the chemists and chemical engineers, that this could never work. So I thought to myself, I wonder if anybody ever wrote anything down. So you can do something called a science citation search. You can look back at your 1976 Nature paper, that's ours, and see who wrote about it. And so I found various different things that were actually very useful, and I'll just show you one. This is from 5 of the, I did this in 82. This is a paper in 79, referring back to our 76 paper. And these were 5 of the leading polymer chemists in the world at the time. I'll just read you what they wrote. They wrote, generally, the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example, proteins, could be released by such a technique because of their extremely small permeation rates through polymers. However, Folkman and myself have reported some surprising, see, that's a very good word for a patent lawyer, results that clearly demonstrate the opposite. So I showed this to my lawyer, the hospital's lawyer, and he said, that's terrific. He flew down to Washington to see the patent examiner, and the patent examiner said, he said, gee, I had no idea. He said, I'll tell you what, I will allow this patent if Dr. Langer can get written affidavits, that's written documents, from each of these 5 people that they really wrote that. This is actually a true story. So I wrote all of them, and each of them was nice enough to write me back that they really did write that. And we got this very broad patent, pretty much any polymer and any molecule. And through that, we licensed it to companies. We taught companies. I even got involved in starting companies. And now there's all kinds of products, based on this on the market. And it's critical for molecules, like I say, that are large and are destroyed immediately. You couldn't use them otherwise. Also ends up being critical for, a lot of times in mental health diseases, people don't take their drugs. The compliance is terrible. They've been used everywhere from prostate cancer, endometriosis. This actually is one of the widest treatments in the world today for schizophrenia, a mental health disease. About 5 million patients have used it. I don't know if people saw the movie A Beautiful Mind with Russell Crowe, but he had schizophrenia. They gave him electric shock treatments. Now he'd get injections of this probably every few weeks. This is actually a treatment they can give, a once-a-month treatment for that can help people that are narcotic addicts or alcoholics and stuff like that. So there's really ends up affecting many, many people. So, so this is one story I wanted to tell you. The second story I want to tell you also, very different origins. So we do a lot in materials in our lab, but when I started, and this is kind of how I got started on materials, so I started to think one day, how do materials find their way into medicine? Is it by chemists or chemical engineers? When I looked into this, I found that was never the case. Pretty much every time a material finds its way into medicine, it's a medical doctor who urgently wants to solve a medical problem. And what they do is they go to their house to find something that kind of resembles the organ or tissue they want to fix, and then they use it in the body. So, for example, in the case of the artificial heart, what they ended up picking was a lady's girdle material because it has a good flex life. But it doesn't work very well. When blood hits the surface of the artificial heart, it forms a clot, and the clot goes to the patient's brain, they get a stroke, and they die. But again, if you think about it, something designed to be a lady's girdle may not be the optimal blood contacting material. And this problem, see, once you start out with a material like this, it's very hard to change it because of regulatory authorities like FDA and so forth. So basically, pretty much every material in medicine today has an origin like this, like dialysis tubing with sausage casing, vascular graft, that's an artificial blood vessel, was a surgeon going to a clothes store to see what he could sew well with. Breast implants, one was actually a mattress stuffing. So I started to think that, we ought to be able to do better. And one of the things you learn in chemical engineering is kind of what I call chemical engineering design. So I thought, why can't we ask the question, what do we really want in a biomaterial from an engineering standpoint, chemistry standpoint, and biology standpoint, and see if we could totally change that paradigm. And to start with this, I picked an example. When I started, the only material that was FDA approved that was degradable and synthetic were polyesters, like sutures. They dissolve like this. It's called bulk erosion. They start out looking like this. They get spongy, and they fall apart, which means you could dump the drug out, which is okay for some drugs, but if you had a toxic drug, like an anti-cancer drug or insulin, that would be bad. So we said from an engineering standpoint, what you really want is you don't want it to dissolve like bulk erosion. What you'd really like is this. How could you do it? So here's just a very generic structure of a polymer, which are monomers connected together by bonds. And what I started to do was just ask different questions. The first question is if I want to get this kind of surface erosion, what should the nature of the monomers be? And we'd say, well, they should be very hydrophobic. and so forth. But actually, let me just even take a step back. The first question you probably want to ask is, if you're going to make something that's degradable, what should cause the degradation? Should it be water? Should it be an enzyme or something else? And so our thinking was, since we're going to design it from scratch, all of you probably have different enzyme levels, but you all have excess water. So everybody has excess water. If we want to make this reproducible, let's build into the polymer the ability to be degraded by water. So that was the first thing. The second thing we said is, if we do that, then if it's going to be surface erosion, we want to make these monomers very hydrophobic to keep the water out. Then the third question we'd say is, if we do that, what should the nature of the bonds be? Because when water does hit this, we want it to dissolve quickly enough. So this comes from organic chemistry. You could look at bond lability from very stable to very unstable, and we chose the anhydride bond. And so this was our idea of synthesizing hydrophobic polyanhydrides. And then the last question was actually building up from scratch. What should the building blocks of these polymers be? Here I asked Michael Marletta. Mike is now head of Scripps, but he was a young professor at MIT then, to help me on this. And we picked different monomers that he thought would be safe. And then we synthesized. So this is what we call a copolymer. This is very hydrophobic, and this is a little less so. And when we made it, what was exciting is not only did these come close to surface erosion, but by just changing the ratios of these different systems, we could make them last. This is like 79% of the one. This goes away in 2 weeks. This will take 3 or 4 years. So you could simply dial in the ratios of these and make these last as long as you want. So one of the things, again, that I mentioned that we wanted to do, and now I'll go back to the brain, is to see if we could help people with different diseases. And Henry Brem was a young neurosurgeon at Johns Hopkins in the 1980s, and he came to see me. He's now chief of neurosurgery there, and he wanted to have a better way to treat glioblastoma multiforme, which is the most common form of brain cancer, and it's uniformly fatal. The drug that they normally used to treat it at that time was BCNU. It's a very toxic drug. And what Henry asked me, and we talked about this, is could we again come up with a different kind of paradigm for treating disease? Could we allow the neurosurgeon, rather than infuse the drug throughout the whole body, to do what we call local delivery? Could you line the surgical so Henry's going to, or any neurosurgeon would operate on the patient, they'd take as much of the tumor out as possible, and then they'd line the surgical cavity with this polymer. That was the idea. Normally it lasts 12 minutes, but if you put it in the polymer, it's protected. And we could make the polymer last for whatever length of time he wanted. So we picked a month from what they did in animals, and so now it lasts much longer. Polymer protects it from degradation, and you want it to be degradable so it doesn't accumulate in the brain, to be surface degradable so it can't dump the drug. And see, the other thing that's very important is high concentrations in the brain where you want it to be, but low concentrations in the rest of the body where it causes harm. Now, one of the things is if you come up with a new, at least in the United States, and maybe here, if you come up with a new idea, and you're in academics, you always have to raise money. And the way we raise money is we write grants to the federal government, and then they have, like this is to what's called NIH, and then they have what are called these study sections, which are professors at other universities, and they review what we do and tell us whether we should get the money or not. So that's what I did, but we did terribly. I'm just going to show you this. I entitled this slide, "This Approach Will Not Work Because." In 1981, when we first proposed it, the chemists looked at it and said, well, we'll never be able to synthesize the polymers. But I had a very good graduate student named Howie Rosen. Howie, by the way, later became president of that Alzic company. It was a $12 billion corporation. Also was elected to the National Academy of Engineering, which is a very, very high honor in the United States, and he synthesized them. So we sent the grants back, and the reviewer said, well, we still shouldn't fund it because the polymers, they have anhydride bonds, they'll react with whatever you put in. But I had another couple postdocs, Cam Leung, who's now the James Duke professor at Duke University, also elected to the National Academy of Engineering, and Bob Linhart, who's Constellation Professor of Chemistry at RPI, and they showed that didn't happen. Sent the grant back again, and the reviewer said, look, it still won't work. By the way, the reason the years take every 2 years is the United States government's pretty slow, too. But at any rate, then the reviewer said, well, it still won't work because the polymers are fragile. They'll break in the body. But I had another couple postdocs, Edith Mathiowicz, who's a full professor at Brown University now, a member of the National Academy of Inventors, and Avi Dome, who later became head of medicinal chemistry at Hebrew University, and they made polymers very strong so that they didn't break at all. Then the next thing is people said, well, new polymers are certainly going to be toxic in the body, so we still shouldn't fund it. But I had another graduate student, Cato Lorenzen, who later became Dean of Medicine at the University of Connecticut, was elected to the National Academy of Engineering and the Institute of Medicine, the National Academy of Sciences, and he showed, which is what Marletta had predicted, that they would be safe. Anyhow, this kept going on and on until 1996, when the FDA approved it. It was the first time in 20 years the FDA approved a new treatment for brain cancer, and the first time they ever approved this idea of local chemotherapy. You can probably tell from the way I'm speaking that I'm very proud of how well all the MIT people did. You know, they became tremendous honors, head of major corporations, held endowed chairs, department heads, and so forth. Whereas the reviewers at the other universities, they didn't do so well. I do want to show you what this operation looks like, but if anybody's none of the people here or any of the reviewers, but but anyhow. I do want to show you what this operation looks like, but if anybody's squeamish and doesn't like the sight of blood, don't look. But these are some human brains where you put the little wafers in. Usually they put 6 or 7 in and close up the brain. You know, it's very hard to get good advice when you give a talk, but about 14 years ago I was lecturing at MIT, and my wife, who I mentioned before, came to the lecture. And at the end of the lecture I said to her, I said, Laura, what did you think of the lecture? And she said, well, Bob, she said, the talk was all right. That, by the way, is incredibly high praise. But she said, you know, you had those 2 bloody slides on. Yeah, I was lecturing to chemical engineers and chemists. And she said, you had those 2 bloody slides on for about 10 minutes where you explained every detail of the operation. She said, all those poor engineers were turning green. So ever after that, I did what I did today. I showed them really quickly. I gave a warning. But just to tell you the sequel to that was about 2 years later, I got asked to give this dinner speech. But this speech was to a group of neurosurgeons. And I told them the same thing. And at the end of the talk, a bunch of the neurosurgeons came up to me and they said, you know those bloody slides you showed? I said, yes. They said, no problem. You could have kept those on as long as you want. They said, but those chemical formulas. You know, to leave those on for a neurosurgeon, I guess, is kind of cruel. At any rate, this is some of the data from what's called a phase 3 trial, actually done here and in Finland. So it was published in Neurosurgery a number of years ago. And what it shows, it's not a cure by any means, but it does prolong. You get a 5-fold increase in survival, and it prolongs life and relieves suffering. It's actually been approved in over 30 countries. It's been used for the last 18 years and really created a whole new paradigm for localized controlled release, which is now used in drug-eluting stents and many other things. You know, I thought, again, particularly this audience, you know, and you have to forgive me, I was thinking, if I was a student, what would also be good to see, you know. So there's various milestones that you could achieve, you know, if you're a professor, you know, you could have a paper accepted in a good journal, you could get a grant, you know, you get promoted. But one thing actually that I never thought about was actually having your stuff featured on American television. And so I haven't shown this in years, but there's this TV show, I don't know if anybody ever saw it, probably a lot of you are too young, in the United States called ER. It was actually the emergency room. It was actually the #1 ranked show in a while. And it was really, I don't watch it, but my sister and a lot of the people in my lab called me up. They said, do you know they actually talked about this stuff on the show? So I thought I'd show you a clip from ER. I hope it works. And so here it is. Oh, but the background is he has a brain tumor. Can we turn it up?
What did he say?
I may go to trial.
Thought you were going to be able to settle.
Yeah, so did I.
They're both doctors.
Rachel called. She wants to come out a day early for Christmas.
Mark Green? Dr. Burke, nice to meet you.
You too.
And you are?
Dr. Elizabeth Corday, Dr. Green's fiancée.
Oh, congratulations.
Thank you.
Sorry to keep you waiting. I met some resistance in Tumor Board.
Is that good or bad?
The oncologists and radiation therapists are having trouble embracing the future.
Meaning?
Come over here. They see a lesion like yours, they jump to the conclusion it's inoperable.
But it's not.
Not in my hands.
He went to Harvard.
There's your tumor. Now, as you were doing verbal tasks, there was increased blood flow to language centers. Those light up in orange. As you can see, while the tumor's adjacent to Broca's, it hasn't invaded yet.
That's what the neurosurgeon in Chicago said.
That's why you're here.
So you'll operate?
Yeah. We'll do an awake craniotomy.
He's going to
You'll be
chemotherapy
conscious
away.
and talking while I map out Broca's and resect the tumor. But The real bonus is my ability to insert high-dosage chemotherapy wafers into the tumor cavity and use the malignant cells themselves to create a cancer vaccine.
Thank God for second opinions.
Yeah, those idiots in the tumor board are satisfied with 12-month survival, where most of the patients on my protocol are disease-free for 24 months and counting.
So, when can you fit him into the schedule?
How about December 31st? You can ring in the new year with a load off your mind. Brain surgeon humor.
Right.
You okay?
Yeah. It's good news.
It's very good news. You came to the right place, Mark.
So I thought I'd give you one last fast example of what we've tried to do. Actually, should we do questions? Whatever you... I'll do it in 3 or 4 minutes. I'll try. So another idea I had once, I was watching a TV show on how they made microchips in the computer industry, and I thought, why couldn't we create drug delivery systems out of them? So we did create these chips. This was done by John Santini, Prasad Shastri, and Mike Seema, who was my collaborator. And the idea is we could put different drugs in these, cover them with the metal. And we made them, actually. This is a picture of a chip. It involves a variety of new chemistries. And we have 34 wells in the top, 34 in the bottom. This is like a short, flat chip. This is a United States dime. But they could be made into any size and shape. And just to show you how they work, here's a well. It's covered, in this case, with gold. And it'll stay like this for years. But you can apply a volt by remote control. And what you'll see in nanoseconds is off. When you apply that, like with something like this, the cover comes off and whatever's underneath it can come out. So you could put different amounts of drug in different wells. This is from another Nature paper we wrote, and you see the drug coming out. Or you could literally have like a pharmacy on a chip. You could have multiple drugs, and they could be programmed to come out at any time. And we actually have now taken this even into humans. This is going to sound like space-age medicine, but we actually did it. We actually worked with a group in Denmark, and you can put the chips in the body and communicate with them over a special radio frequency called the Medical Implant Communication Service Band, and it's approved by various authorities. You even have a special code you can put in so only the doctor or patient can control it if you want. And then you have this bidirectional communications link, which could actually be a cell phone even or something like this, and you could confirm whatever happened. The trial we did was 8 patients. We wanted to pick a really hard drug because people would say the reason this wouldn't work is you get this fibrous encapsulation and large molecules can't come out. So we actually picked parathyroid hormone, a fairly large peptide. We also chose it because we felt we could make a significant clinical impact because 77% of the women who take this, they take it for osteoporosis. They don't keep doing it. They just drop out. So we felt here it's going to do it itself over and over again. This was just done in a small procedure in the doctor's office, and actually we got the same pharmacokinetics with less variability, and 3 key measures of calcification of whether you're treating osteoporosis were the same as daily injections. And here's just some of the human data. This is the release rates in the blood at days 60, 68, 76, and 84. It's pretty much superimposable. Here's actually what it looks like. It's actually a little small system. There's a chip, then there's a power source, microelectronics, and a computer program built into it. And on the other side, which you can't see, there's actually an antenna imprinted on it, and that's how you communicate with it. We do get some fibrous encapsulation, but not enough to block anything, and it's about 1/40th of what a pacemaker gets, and no inflammatory cells is judged by histology. We're now taking this into about 4 directions, and I'll mention each of them very quickly. The first direction is making a 2-year device for this. The second direction actually was kind of interesting. I didn't think of this myself, but I had a visitor about 2 years ago. This is me. You may recognize this gentleman. If you don't, he's been responsible for a lot of the computer technology we have. And he has a big foundation. And he and his wife asked, right now when people have birth control, once they put it in, they can't control it. And they want for the third world, it's Bill Gates in case people don't know, but what they want for the third world is the ability that a woman could turn it on or off any time. And you can't do that right now with any birth control. But with this, you can. So they've actually now given us over $6 million to create a personal fertility control system with emphasis for use by women in the developing world, so that they can regulate their own fertility. The third thing that we're doing, actually, we've gotten grants for with Anne Graybiel, who's a very good neuroscientist at MIT, very famous neuroscientist, was actually my wife's PhD advisor, and we're actually now implanting it in the brain to try to have a new device. These are the titles of the grants: for electrical and chemical modulation of pathologic neural activity and devices for traumatic brain injury. Specifically, what we're doing is we're making tiny, we actually call them injectrodes. They're about 250 or 150 micrometers, but they can actually record and even stimulate neural activity and simultaneously deliver multiple drugs into whatever region we implant it. So we're just starting this, so I just thought I would share it with you. We're looking at drugs that can increase or decrease excitability, neuromodulators, drugs that could be used in treatment of anxiety and mood disorders, and the long-range goal is a closed feedback mechanism to control delivery of therapeutics for only when the patient needs it. So some of the things that we've already done are look at, this is now in monkey brains, which Anne is an expert at, ways of silencing neurons, and you can even put tracers in and look at the diffusional spread in the rat striatum. So I'm going to stop there. Our long-term goal for so many of these things is actually we can even put sensors in these chips, so someday you could actually, whether it's for brain or other things, you could actually even sense what's going in the body and then maybe have the chip in turn deliver something in response to a signal. Anyhow, I want to thank you very much for having me. It's been an honor to speak to you. Thank you.